Hello, my fellow biologists. I'm so glad you decided to come back for another biology lesson. Grab your guided note packet, a writing utensil, and let's jump right in because it is time for. We'll be starting off our Unit 4 with Lesson 1, which deals with DNA structure and function. In order to understand the concepts that are going to be covered throughout the entire unit, we need to understand the basics first, like what DNA is and how it works. DNA itself is a three-letter acronym that stands for deoxyribonucleic acid. DNA is also the genetic material responsible for all traits of living organisms. Traits are those defining characteristics that make us us. DNA is responsible for coding for those traits so that we can exhibit them. DNA is located within all eukaryotic organisms inside the nucleus. Remember, the nucleus is the organelle inside eukaryotic cells that protects the DNA. In prokaryotic cells, DNA is just floating around inside the cytoplasm. To understand DNA a little better, we first need to understand the components or what makes up DNA. DNA itself is a long polymer composed of many nucleotides. A polymer in biology or chemistry is nothing more than a very large molecule that is made up by combining smaller molecules. We make polymers by combining smaller molecules, and when it comes to DNA, the molecules that we are combining make this very large molecule called a nucleotide. Now, nucleotides are made up of three things. We have our nitrogenous base, a phosphate group, and a deoxyribose sugar. I'm going to repeat that one more time. The three things that make up a nucleotide are a nitrogenous base, a phosphate group, and a deoxyribose sugar. Now, if you take a look at your screen, you will see two images. The images on your right show what a molecule looks like as well as the labels. I want you to take some time to label the image in your notes, matching it to the image that you see on the screen. If you need to pause the screen, that is fine and highly encouraged. On the left side, you will see the nucleotides combining with each other to form the large DNA polymer molecule that we talked about. One part of a nucleotide is its nitrogenous base. Nitrogenous bases are pretty important because they are the part of the nucleotide that carry the genetic information. They are responsible for carrying those traits that we are going to express later on. Now, there are four different bases found in DNA. The bases are adenine, cytosine, guanine, and thymine. Now, one more time, the four nitrogenous bases are adenine, cytosine, guanine, and thymine, all found in DNA. Now, those nitrogenous bases are capable of pairing with another nitrogenous base on a complementary strand of DNA by hydrogen bonds. So if you're looking at the image on the screen, you're going to see that we have adenine, which is a nitrogenous base, pairing itself with thymine. Then if you look below the adenine and thymine pairing, we have guanine pairing itself with cytosine. This keeps going on and on and on and on and on and on until we get this very large DNA molecule. You may be wondering how these bases know their pairs. There was actually a scientist who did an experiment that solved this problem. The scientist by the name of Chargaff mixed DNA with an acid so that the nucleotides would separate. He observed that the nucleotides traveled up the piece of paper, and they did that based on the size. He noticed that the bigger nucleotides traveled at a slower rate than the smaller nucleotides. They traveled a little bit faster. From this, he was able to infer that the amount of adenine was equal to the amount of thymine because their levels were the same on the paper, or that they essentially traveled at the same rate. And he saw the same thing in cytosine and guanine. They were equal on the paper as well. From this information, he was able to get the base pairing rule. The base pairing rule is when adenine is going to equal thymine and cytosine will equal guanine. An example of this would be, I have a problem in which I have 18% adenine. There should also be 18% thymine because they are equal to each other. And if I have 35% cytosine, I should also have 35% guanine. Now let's touch base on DNA shape. We should know that DNA is a double-stranded molecule, meaning that it is made up of two strands. These strands are connected by the nitrogenous bases pairing with each other. Purines are larger nitrogenous bases, and purines are adenine and guanine. Pyrimidines are smaller nitrogenous bases, and those are thymine and cytosine. When the nitrogenous bases pair correctly, the width of DNA stays uniform throughout, and this is what's helping us create that polymer that we talked about earlier. Let's have a look at the images on our screen. On the left, we see our category of purines, which are adenine and guanine, and they combine with our pyrimidines, thymine, and cytosine. Now remember, the way that they combine are based on Chargaff's rule. Adenine combines with thymine and guanine combines with cytosine. Now let's take a look at the image on our right. We see that two purines were to be combined with each other. They would create a molecule that's too wide. Two pyrimidines combining together with each other, it would create a too narrow strand or too small, but a purine and a pyrimidine combining with each other makes the perfect fit. When it comes to DNA, we know that nitrogenous bases are held together by hydrogen bonds. Hydrogen bonds are relatively weak, and that's going to be important in our later lessons. The sugar and phosphate that we talked about that make up DNA are held together by covalent bonds, which are relatively strong. This creates a sugar-phosphate backbone with the nitrogenous bases on the inside. DNA is typically wound up in a twisted ladder, which you guys will refer to as a helix shape. It kind of looks like a winding staircase. DNA itself is arguably one of the most important molecules on the face of the earth, and that is because DNA itself is the blueprint for the different proteins in the body. Proteins are important because they are required for structure, function, and the regulation of all activities within the cell. To get a better idea about this, we should know that DNA has genes on it, and genes are the coding segment of DNA that is specific recipe for a specific protein. DNA has genes, and these genes call for proteins. And these proteins are going to in turn carry out work of the cell. Dependent upon the work that is done in the cell is going to lead to the characteristics that we express or what makes us unique. Well, ladies and gents, that is the end of our lesson for today, so please continue to do the practice that is at the end of this lesson. Now, you need to come to the next class with this complete. If you need to, please go back through and utilize the notes that we just took to complete the practice. Now, if you have any questions, please know that you can stop by and check in with myself, Mr. Brown, or Miss Ray. Have a great day.